In the past few classes, we have completed the part where we discussed the raw materials required for the translation machinery. We studied that the ribosome translates the genetic message from mRNA into proteins. The mRNA is translated from the 5' to 3' direction, producing a corresponding N-terminal to C-terminal polypeptide. Amino acids bound to tRNAs are inserted in the proper sequence due to, first, specific binding of each amino acid to its tRNA. Second, by specific base pairing between the mRNA codon and the tRNA codon. So we have learned this. Now we are coming to the phase where the translation machinery is operative. So if we look into the prokaryotic and the eukaryotic translation machinery, it is in both cases, it is divided into an initiation, then we have an elongation, and then we have a termination. So during initiation, as you can see, the ribosome binds with the mRNA, then gradually, and the initiation complex is formed. We'll be getting into that. Then as the initiation is finished and elongation process, there are successive cycles of the amino acid tRNA binding and peptide bond formation until the ribosome reaches the stop codon, and finally termination when the ribosome reaches a stop codon, the mRNA and the protein dissociate from the ribosome, and the translation for that particular mRNA is completed once. That is one cycle of translation. So we have an initiation, we have an elongation, and we have a termination phase. The remaining I'll be explaining with you via. So as I just now said, translation is a process whereby the base sequence of a messenger mRNA is converted into the amino acid sequence of a polypeptide. This process involves an mRNA, a tRNA, amino acids, and ribosome, and some accessory proteins. We have done this. This is still a recapitulation. The mRNA consists of three main parts. The portion from the 5' end of the translation start codon is the 5' untranslated leader sequence. Then we have the open reading frame, the central portion. It commences with the AUG start codon, followed by a series of 3 nucleotide codon, each of which specifies one amino acid. And finally, it ends with a stop codon. There are 3 stop codons, UAA, UAG, and UGA. The final portion of the mRNA consists of the section called the 3' untranslated trailer sequence. The amino acids vary in length because of the length of the 3 different parts. So that is an mRNA. We have discussed the tRNA also. The tRNA brings amino acids to the growing peptide chain during translation. It consists of a single RNA chain of about 75 to 90 nucleotides. Each tRNA have similar base sequences. After it is transcribed, as you can see, the tRNA folds into a characteristic cloverleaf model. The anticodon sequence is the 3 nucleotide sequence that is complementary to the codon of an mRNA. The amino acid is attached to the 3' end of this cloverleaf. Coming to the ribosome. The ribosome assembles during translation, and it codes the mRNA into a polypeptide. It consists of 2 parts, the larger subunit and the smaller subunit. And as you can see, the active ribosome, we discussed the other day, consists of 3 binding sites. The A site, the P site, sorry, this is the A site, the P site, and the E site. So here is the E site, the peptidyl site, and the amino acid site. In prokaryotes, translation initiation occurs in 3 basic steps. As you can see, this is the 30th ribosomal subunit, and in the next moment you will see that the 30th ribosomal subunit binds with the initiation factors FN1, initiation factor 1, initiation factor 2, and initiation factor 3. Initiation factor 2, in fact, can bind with the GTP, and it is the breakdown of the GTP that provides transient energy for the process to start on. So whenever there is a GTPase, IF2 is a GTPase, that means it can bind to a GTP molecule and its functional efficacy is enhanced when that GTP is broken down to give GDP and release of phosphate. So let's see it. So you can see it's getting attached to the 3 initiation factors. And then, as I said, IF2 is actually the GTPase. So it's binding with the GTP. And now we had studied that upstream in the 5' untranslated leader region, there is a sequence which is known as the Shine-Dalgarno sequence. We have studied this. This is a purine-rich sequence. And the ribosome with the IF initiation factor GTP complex will recognize this Shine-Dalgarno upstream of the UG start codon. As I had earlier stated, this is called the ribosome binding site. This sequence is complementary to a sequence at the 3' end of the 16S ribosomal rRNA of the 30S ribosomal subunit. And it serves to align the ribosome on the mRNA in its proper reading frame. So let's see that. So that's the Shine-Dalgarno sequence. And if you remember, it's a purine sequence AGGAG. And it's complementary to the pyrimidine-rich sequence in the 16S ribosomal rRNA. Now, the second part, so we see the first part is the attachment of the ribosome to the mRNA. Now, so the ribosome is positioned, the ribosome has aligned itself. Now, the second part is for the translation to begin, the tRNA should also come. Now, we have this start codon, a few nucleotides downstream of this Shine-Dalgarno sequence. One thing to be noted is that AUG is the initiation codon, and that codes for methionine. In case of prokaryotes, it has been seen that the initiator methionine, that is the first methionine to be attached to, or the first methionine making a polypeptide, is a bit modified form of methionine. In this methionine, the modification that occurs is the addition of a formyl group to methionine, making it formyl methionine. So let's see that. So this is the initiator tRNA with the anticodon of methionine. And we have studied the charging of the tRNA, and methionine has been added. But this methionine is modified. You can see a formyl group is being attached to the methionine. And this formyl methionine is now being carried by this initiator tRNA. And you can see the initiator tRNA with the formyl methionine is hydrogen bonded with the AUG codon. And the moment it binds, you can see, you saw there is one change. The IF3 is released with the remaining assembled complex. So the IF3 is released. And this remaining complex is called the 30S initiation complex. Let's go down a bit behind and see. So C is joining and the IF3 is released. And we are seeing the 30S initiation complex. So, however, the initiation process is not yet complete. The 70S ribosome, sorry, the 50S large ribosomal subunit will now bind to the 30S initiation complex. And the moment this happens, as I said, there is a GTP bounded. So the GTP is hydrolyzed and IF1 and IF2 are released. And the final complex will be known as the 70S initiation complex. Also, when I start the animation, note that there are 3 pockets in the ribosome as discussed earlier. So we have a P site, we have an A site, we have an E site. Remember that the initiator tRNA is situated at the position of the P site when the 50S ribosome binds with the 30S ribosome. So let's go over the animation. So you can see the 50S ribosome sitting. And as I said, you can see that the initiator tRNA is sitting in at the 50S, the initiator tRNA is sitting at the P site of the 50S ribosomal subunit. And there's a breakdown of GTP with the release of the initiation factors. So now we have this 70S initiation complex ready. And now the ribosome is positioned on the mRNA in its proper orientation. And we have a situation where we have an empty A site and we have an empty E site. In the empty E site, as you can see, there are 3 codons now to be read by another set of tRNA carrying a specific amino acid. So we have our 70S initiation complex ready to proceed for elongation. So once the initiation process is completed, as we saw in the earlier animation, the translation elongation phase begins. Now, the elongation phase consists of 3 steps. The first, as you can see, if you look into this particular structure, we have a mRNA, we have the initiator tRNA position in the P site, we have an empty vacant A site, we have an empty vacant E site. And there's another droplet, as you can see on the background, that is actually the enzymatic center of the ribosome where the peptide bond formation will take place. Now, as I said, if we look into the elongation phase, it consists of 3 basic steps. The first step is the binding of the tRNA to the ribosome, formation of the peptide bond between adjacent amino acids, and the movement of the ribosome to the next codon. Elongation phase, as you will see, will continue 1 codon at a time until a stop codon is reached. So let's get into that. So it consists of 3 phases. And we have a new codon being present in the A site, as you can see from this picture. And this codes for serine. This particular codon codes for serine. So when the initiation concluded, and we saw that the F-met tRNA was hydrogen bonded to the AUG site, to the AUG in the P site, the next codon in the A site, the A site is empty and we have our next codon in the A site. Keep it in mind that codon means 3 nucleotide bases. Now the UCC codes for serine. So it's obvious that serine bearing tRNA in the cytoplasm will now come. While we'll be seeing this elongation phase, we'll see that just like in initiation, there were certain proteins which helped in the initiation machinery, if you remember, initiation factors. Similarly, we have certain proteins which also play a big role in the elongation. Now, this protein is the elongation factor TU GTP complex. So let's see that how that happens. Let's go over it. So this is the serine tRNA. And you just look at the colors. This is elongation factor TU. This is the GTP form. This is the GDP form. And this is the elongation factor G. Now, I'll just concentrate here. What did we see? That this is a tRNA being charged with serine. This is now moving towards the A site. When it is moving towards the A site, it gets itself attached to EFU, TU, and GTP. You can see this EF, TU, GTP will help it to enter into the A site. So let's see. So it's entering And once hydrogen bonding occurs, you'll be seeing a breakdown of the GTP with the release of the EFU, EFTU, and the GDP form. So just once more, what did we see? Just a second. We saw the EFTU GTP bond form. Once hydrogen bond is completed, we are seeing that the GTP is breaking down and EFTU is released. Now you see in the P site, we have the initiator tRNA with the first amino acid, which is a modified form of methionine, formal methionine. And the A site, we have now a charged tRNA carrying serine. So the next step will be, as you will see, is the formation of a peptide bond between the carboxyl group of the methionine and the amino group of serine. The peptide bond formation is a simple condensation reaction with the liberation of water. So that is what we'll be seeing in the next step. So our P site and A sites are now filled. So as I said, there was this droplet which is now surrounded by yellow halo. You can see this reaction is catalyzed by peptidyl transferase residing in the large ribosomal subunit. The first step involves the breaking of the bond between the methionine F-met, the formal methionine, and the tRNA in the P site. So you will see once I start the animation, this bond will be broken. And next you will see is the peptide bond formation between formal methionine and serine, which is attached to the A site. So we'll see a bond breaking here at this end. So the bond will be broken here first, and the peptide bond formation will take place here. So let's see that. So you saw? So let's get over it once more. See, the bond is broken, and then we are seeing the peptide bond formation between formal methionine and serine. This peptide bond formation is catalyzed by the enzyme peptidyl transferase. So now, before getting into that part, let us talk about this peptidyl transferase. Now, this peptidyl transferase initially was thought to be an enzymatic protein, but recently it has been shown that this peptidyl transferase is nothing but a catalytic RNA molecule. We studied such catalytic RNA molecule when we did the spliceosome machinery during transcription. And if you remember, I had said that these catalytic RNAs are known as ribozyme. So in the 50S ribosomal subunit, we also have another catalytic RNA, which is known as the ribozyme, which is sitting here and is facilitating the bond formation between the formal methionine and the serine. So it is an RNA molecule that is helping in formation of the peptide bond between the formal methionine and serine, and such an RNA is known as the ribozyme. So you can see this is there will be a condensation reaction. First, this bond will break. And as I said, there will be a condensation reaction between the carboxyl group of methionine and the amino group of serine with the liberation of water. So you saw the peptide bond formation. Now, look at the position of the ribozyme. We have a P site, which is now containing a tRNA whose amino acid has been removed. We now have an E site, which is empty. And we now have an amino acid site, which is now containing the growing polypeptide. So the next thing that happens is that once the peptide bond is formed and there is an uncharged tRNA in the P site, and a tRNA with a growing polypeptide chain in the A site, the ribosome will now move one codon. It will now move one codon. This movement of ribosome on the mRNA by one codon is known as translocation. When the ribosome will translocate, it again needs the help of certain proteins which will help in its translocation. And you can see this blue color ball, this is the elongation factor G, which will help. This can also bind with the GTP, and with the breakdown of GTP, the energy released will help the ribosome to move one codon over the mRNA. So when the ribosome will move one codon over the mRNA, you can assume that the uncharged tRNA will move into the E site. The tRNA containing the growing polypeptide will move into the P site, and we will have a vacate empty A site. So elongation will now start. You can see the elongation factor G binds with the GTP. You can see it has got attached breakdown of GTP to GTP, and you can see the ribosome has started moving. Okay, so as I said, the moment the ribosome moves one codon, we have the uncharged tRNA on the E site, the peptide, the tRNA with the growing polypeptide on the A site, P site, and we now have again a vacant A site. So this A site is now again open for the entry of a new tRNA. And one more thing which we observed just now is that the moment we have an uncharged tRNA on the E site, the E site exists from the ribosome. Now the A site is now open for a next round of amino acid tRNA to enter and the elongation process to continue. So let's go over it once more. Now, as we saw that initiation occurred and then elongation started, and elongation will continue as long as the A site of the ribosome doesn't reach a stop codon. The moment the A site of the ribosome reaches a stop codon, then there is a different machinery that comes into play. So if you remember a class of genetic code, we have these three stop codons which do not code for any amino acid. So let's get into that. And thus, as they do not code for any amino acids, so tRNA have anticodons. tRNA do not have any anticodons for this. So these are the standard stop codons, which is universal in nature to some extent. There are exceptions. So this is where we had seen. So if you can see, what did we see? So this is the E site, this is the P site, and this was the A site containing the growing polypeptide, tRNA containing the growing polypeptide. As we saw how the amino acids are being added. So one after another, this was how the amino acids were added. Now, in the next phase of translocation, the moment the ribosome will now move one codon, as we studied in the earlier animation, you will see that this tRNA with the growing polypeptide will move into the P site, and the uncharged tRNA will move into the E site. And in this vacant A site, we will now have this stop codon. This stop codon can release, this stop codon can be recognized by three categories of release factors. As I said, they do not have any cognate tRNA, but these Stop codons can be recognized by these three categories of release factors. RF1. So let's get into that animation first. So these are the release factors. Let's see what's happening. So we have RF1, release factor 1, release factor 2, release factor 3. You can see release factor 1 can recognize UAG, release factor 2 can recognize UAA, UGA, and release factor 3. Let's get over that once more. Let's see the properties once more. So release factor 1, UAA, UAG. Release factor 2, UAA, UGA. Release factor 3 does not recognize any stop codon, but stimulates the subsequent process of termination events. Now, translocation occurs, as I said, and we now have this stop codon on the A site. Release factor 1 recognizes the stop codon. And as you can see, once the release factor 1 recognizes that stop codon, what did you see? Once the release factor 1 recognizes the stop codon, this peptidyl transferase will now cleave the polypeptide that was on this tRNA. You saw it? See. So the peptidyl transferase will now cleave the polypeptide. So the polypeptide has left. Now the tRNA is then displaced from the P side, followed by the disengagement of the release factors. And finally, as you can see, the 30S and the 50S subunits open up from the mRNA. So this is the termination of translation. So as you can see, the ribosome binds at the ribosome binding site, then recognizes the AUG, initiation starts, and elongation continues. And on reaching the stop codon, the ribosome opens up. So as you can see, this is the movement of ribosome. So the ribosome is moving from the 5' to the 3' direction. And the polypeptide is being synthesized from the N-terminal end to the C-terminal end. So you can see that at a given point, once the first ribosome moves away from the RBS and enters into the elongation phase, a second ribosome can bind to the RBS, form the initiation complex, and can again enter the elongation phase. A third ribosome can bind again at the RBS, start the initiation phase, and can enter the elongation phase. So you can see multiple ribosomes, one after another, with a definite time lag between them, can enter the elongation phase and can rapidly translate the mRNA so that multiple polypeptides of the same mRNA is being produced. So when there is an emergency and requirement of a particular protein, this is how the machinery is fastened or enhanced. And so with this, I come to the end of my translation class.